Hello everyone. Today I'm going to talk about the evolution of non-homologous end joining repair pathway across bacteria. DNA is prone to a number of different kinds of insults, and one of the most lethal of these insults is when there is an attack on the sugar phosphate backbone that can give rise to double-strand breaks. Therefore, one can imagine that it becomes imperative for our organisms to invest in mechanisms that can repair such double-strand breaks. Now, depending on the availability of the template DNA, there are two kinds of repair pathways that exist. The first is called as the recombination-based repair, where a template can be used to carry out the repair. The other kind of repair is when the template is not available, and in doing so, deletions and insertions might get introduced in the gene. This sort of repair is called as non-homologous end joining repair, or NHEJ, and this is what is going to be the topic of my talk as well. Now, in terms of their conservation, what we know is that both these repair pathways are well conserved across all eukaryotic genomes. But surprisingly, a few studies done in the past 2 decades have suggested, based on few genomes, that NHEJ might not be as evolutionarily conserved as recombination-based repair in prokaryotes. This was really fascinating for us, and we set out to ask a very simple question, that was, what does it mean for bacteria like Pseudomonas aeruginosa to harbor NHEJ, whereas organisms like Escherichia coli do not have it? Now, in bacteria, NHEJ is just a two-component machinery. So whenever there is a double-strand break, the first protein to be recruited at the site of damage is called Ku, which then recruits another protein called as ligase D or LigD, which consists of 3 domains: the polymerase, the nuclease, and the ligase domain. Now, the polymerase and nuclease, they do the end processing of the damaged sites and make sure that the 5' phosphate and the 3' hydroxyl moieties are then available for ligase to repair the DNA. So with that in mind, we defined 3 objectives for our study. The first question that we asked was, what is the pattern of occurrence of NHEJ across bacteria? So for that, we took a data set of around 6,000 bacterial genomes, and we carried out a homolog detection based on HMM models. And what we find is that there is a sporadic distribution, there is a patchy distribution of NHEJ domains across the entire bacterial phylogeny, and that this holds true even at the clade level, which suggests that there is a very low phylogenetic signal of NHEJ across bacterial phylogeny. We divided our organisms into different categories based on the presence and absence of Ku and ligase D. There were organisms that coded for neither of these 4 domains. Then there were organisms that coded for domains for Ku or ligase D, which we termed as incomplete NHEJ machinery. And finally, organisms that coded for both Ku and all the 3 domains in different combinations of ligase D. Now, given this pattern, then we asked what evolutionary history could have shaped such a distribution. So for that, we looked at 2 components. The first component that we looked at was the vertical gene transfer component. And what we find is that most likely the eubacterial ancestor was devoid of NHEJ. It was in fact gained multiple times across different lineages independently. But having said that, it was not sufficient to stabilize the trait because we observed multiple secondary losses and gains thereafter. In terms of gaining NHEJ, what we find is that the most common kind of gain was a sequential gain, where ligase D was gained first, followed by Ku during the bacterial evolution. In the reverse direction, the most common kind of loss was a direct loss, where both the proteins were lost at the same step to give an NHEJ deficient organism. Another way of asking the same question is what is the least likely path taken during the evolutionary history? So what we find is that the least amount of time was spent in the Ku-only state, and the transitions to and fro Ku-only state were also the rarest. From studies, we know that Ku has been shown to interfere with recombination-based repair. So what this collectively suggests is that there might be a cost associated with maintaining just Ku alone in the genomes of bacteria. Next, looking at the horizontal component, what we find is that horizontal gene transfer events might have shaped the NHEJ events or distribution, not just among bacterial phyla, but also between archaea and bacteria. And just to give you one example, if you focus on this asterisk over here, which is showing a transfer event between archaea and the ancestor of firmicutes and actinobacteria, we find that when we do a principal component analysis for these Ku sequences, we find that there is a distinct cluster that forms on the bottom left on the principal component 1, which consists of archaea, actinobacteria, and firmicutes, suggesting that specifically these organisms might have participated during the transfer events for NHEJ proteins. And finally, we looked at what evolutionary forces could have contributed to NHEJ evolution. So for that, we took 2 genome characteristics. One was the genome size, and the other was growth rate. Now, both these genome characteristics can be then thought of as being decisive in carrying out recombination-based repair versus non-homologous end joining repair. So what we find is that the organisms that have NHEJ tend to have larger genome sizes as compared to the ones that lack it, and that this is not an artifact of larger genomes coding for more genes. We also find that the organisms that have NHEJ tend to be slower growing as compared to the ones that lack it. However, when we control for phylogeny, we find that this effect still holds true for the association between NHEJ and genome size, but does not hold true anymore for growth rate and NHEJ. We also did a correlated evolution analysis, and what we find is that there is a signal for NHEJ with genome size and NHEJ with growth rate with respect to correlated evolution. However, when we do a phylogenetic logistic regression, what we find is that genome size alone is a better predictor of NHEJ and not growth rate to that much extent. So in summary, what I've told you today is that there is a sporadic distribution of NHEJ across the bacterial phylogeny, and multiple gains and losses might have shaped NHEJ distribution, and how genome size alone is a better predictor of NHEJ state as compared to the other predictor that we looked at, that was growth rate. In sum, what our study brings out is that we can extend this pipeline that we have used in our study to ask similar questions in the evolution of other repair pathways and how they might have impacted the bacterial genome evolution, and how in turn bacterial genome evolution might have impacted how DNA repair systems have ended up in these genomes. You can refer to this QR code for my paper, which is now published in Genome Web Biology and Evolution, and I would be happy to answer any questions that you might have. You can reach out to me on these email IDs. Last but not the least, I would also like to take this opportunity to say that I'm looking for postdoc opportunities as well. So I would be happy to discuss any such advancements. Thank you.